Okay, so this is the written part of exam 3 that I'm just going to go through with you. My name is Steele, and just going to go through this here. So for question 1, this question concerned the human disease that we have discussed in this section of the course. Below is a group of terms that we have learned about recently. So parts A through D here, I just numbered them for myself just to make it a little bit easier in the beginning, but which of the above terms is associated with Patau syndrome? And I got that was a trisomy, and also aneuploidy, and trisomy pretty much falls, not pretty much, it does fall under the aneuploidy, which is like a different number of chromosomes, so you can have trisomy or monosomy that fall under that. So for A, I got trisomy and aneuploidy. For part B, which of the above terms is associated with conventional form of Down syndrome, that's also trisomy, aneuploidy, and I also got a non-disjunction for that. And for part C, which of the above terms is associated with Turner syndrome, which would be 6 and 4, so aneuploidy and monosomy, since missing the Y chromosome on that one. And then for part D, which of the above terms is associated with Kruger-Schott syndrome, and that is simply a deletion. And there's also this little sneaky E right here that I almost missed actually, but which of the above terms is associated with type A hemophilia, and I have for number 2, that's just an inversion. So I have them written here as well as numbered along every one. Okay, examine for number 2, examine the E. coli DNA sequence shown below for each of the primers, and then you have the bottom strand, which is the template strand, and the 3' to 5'. And so for part A, it said indicate the polarity of the primers on the image above. So the polarity I had on these was 5' to 3', 5' to 3', in that order. And for B, what is the DNA sequence that will be synthesized between the primers? Be sure to indicate the polarity. So the DNA sequence between the two primers, so just had to locate the two. So I had primer A matching up right here, primer B matching up right here, so the strand that would be in between them would be just the matching strand for along your template strand, so the G's and the C, C, G, T to the A, so on and so forth. And since the template strand is in the 3' to 5', this will get synthesized 5' to 3', so that's where I have that indicated. And then which direction, right to left or left to right, is the replication fork moving as this daughter strand is being synthesized? And I do have that moving from right to left since it's synthesized 5' to 3'. And then which of these two primers are synthesized first? So if I got C right, I'm moving from right to left. You're just going to run right into that B primer first, and that will be your first synthesized primer. So for C, it says suppose an individual is heterozygous for the following change in chromosome structure, and you have your normal chromosome and then your inverted chromosome, and you have your centromeres here and here, and suppose that these two chromosomes replicate prior to meiosis. The two replicated chromosomes then attempt to synapse during prophase 1. What is the allele and centromere composition of the 4 products of meiosis if there is a crossover between T and U? So I was doing like a translocation cross first here, and I was like thinking something else. I wrote all this stuff out, and then I remembered it was kind of like one of the problems we had done in the practice or in the group, and I just remember that they fell the 4 gametes in this type of a case inversion. One was normal, one was inverted, then you had 2 abnormal chromosomes. So there's a crossover between the T and the U, which I have indicated right here. So basically the first 2 are going to be normal, one's going to be the normal, the second one's going to be the inversion, so those are each. So for the first gamete, you're going to have a normal one with 1 centromere, and that's the standard order for that. And then the second gamete, you're going to have the inverted chromosome, also with 1 centromere, and that's the order, same as it's written up here. And then your third and fourth gamete are abnormal chromosomes, which means at that cross they separate so that the LMN, all caps, kind of like twist down right here, so it's like this whole thing. So this is the one start and then it finishes, or vice versa. The other one is just this little end. So for that first one I did the little ends, so you start XST UVWX with 0 centromeres, and then the fourth one, being abnormal, on the other side it has the double centromere or 2 centromeres, and I have that written out here. So just looping from that top LMN to T, then U to the L again, lowercase there. Okay, and for number 4, it says suppose the DNA molecule shown below is a portion of an E. coli gene. The E. coli gene is transcribed from right to left. The +1 site is shown in bold, which is your TA, and I just had jotted down some things that I thought were important to recognize. I didn't catch anything in the -35 sequence, but I did catch the -10 sequence lining up right here. And these are just kind of notes for me, but which strand, upper lower, is the template strand, and which upper lower is the coding strand? So for this I got that the template strand is the upper strand here, and the DNA and the lower is the coding strand. And what is the sequence of the mRNA molecule produced from this DNA molecule? And basically it's going to start right here where the A is. Since our template strand is the T, the mRNA molecule produced essentially matches this this bottom strand here. And actually, now that I think about it, I think these T's might be U's. So if that matters, then they might be used since it's RNA instead of DNA. So I guess we could go ahead and switch that in our minds here. But that would be, I just indicated it here, 5' to 3' on this side over here. And then in which direction, left or right, along the coding strand relative to the +1 site would the -10 and -35 sequences be located? And I put on the right because I only saw the +10. I didn't see the -10 because I didn't see the -35, and I just figured that with typical sequences it's further along to the right. In which direction, left or right, along the coding strand relative to the +1 site, which is our A right here, would the terminator be located? And since we're transcribing right to left, our terminator, it would make sense that it would be on the left side over here somewhere. So those are the answers that I got for Exam 3 written part.